Okay, I know we have some people who are filing in, but I'm going to get started because I think we have a really packed agenda today. So I'm Stacey Heilman. I'm the Director for Pediatric Research Operations for the Pediatric Institute. And I'll be introducing the speakers today and doing a little bit of housekeeping related to this session. But to those of you who are new to K-Club, welcome. And to those of you who have been many times, we're glad you're back. K-Club is a tiny bit of a misnomer because it's not just for people who are aspiring to get a case. There are some people who already have case here. There are some people who are earlier in their careers. There are some people who are supports to help people and push their research endeavors forward. And so we welcome you all, and we're glad you're here. And if this topic is of interest to you, then you should be here. Before I introduce our speakers, I'm going to go ahead and do a couple of the housekeeping that we normally do at K-Club. So the first, and one of the most important aspects, or not the most important, but a very important aspect of our K-Club is we put this on for you. And so we want to know what you want to hear more about, what maybe is important to you, what that would help you get to that next stage of your career. So we have a survey drawing at the beginning of every K-Club, and the names that are in the Crown Royale bag are the people who responded to our survey from last month. So if you would be so kind, at the end of the session, Barbara Kilborn will send you a link to SurveyMonkey. Should take less than 5 minutes to complete. Give us your feedback. What did you like about today? What do you want to hear more about in the future? What was important to you? So please feel free to please go ahead and respond to that survey, and then you will also be entered into the Crown Royale bag for the limited edition K-Club mug right here. So this could be yours. I'm going to go ahead and have our speaker I'll introduce in a moment. Draw the name from last month. Taylor Mellinson. Is Taylor here? Yep. Oh, congratulations. Thank you for your feedback. Sorry, this is your life. You're coveted. Thank you, Taylor. Okay.
There was no collusion.
No, none. Very rigorous and transparent. Shoot. Hey, Ryan just messed this up. Okay. Or maybe you did? Okay. There we go. I got it. Sorry about that. We have computers going up here, so it's a little confusing.
Yeah, but it's, sorry.
So one other thing, just another housekeeping thing, sometimes in K-Club we'll give just a little shout out to a couple of funding opportunities that we think might be relevant to this audience. So what I wanted to tell you about is an R01 that's specifically designed for early stage investigators, so those who have not competed successfully for R01 funding in the past. So this is called the Physician Scientist Research Award for Early Stage Investigators. This particular one is only sponsored by NHLBI. But if you're doing research in any of the heart, lung, blood diseases, sleep disorders research, this one could be right for you. Protects a minimum of 50% of research effort, and there's a couple of lines coming up, one in June of this year and then next year. So just something to think about. The link is there for that particular one. I also wanted to tell you a little bit about the Paul Foundation Grant and Aid Program. It's just a little bit of money, and it's specific to people who are doing research in basic neurobiology, so very basic science-oriented. But I wanted to point this out because although it's a little bit of money, it kind of supports the concept that we sometimes talk about in K-Club, that it takes money to make money. So it's hard to sort of reach that barrier of entry into the funding game, but once you do, it becomes a little bit easier. So this is an area of research that you are in, you might want to consider that. Okay, so before I introduce the speaker, I just wanted to make sure we were all on the same page about what today's session is about. So you hear these terms for the past couple of years kind of thrown out by NIH. Reproducibility, rigor, transparency. What does it all mean? So I sort of created this color-coded document, and so we're all using the terminology the same way. So I think reproducibility through rigor and transparency The key goal is to enhance reproducibility, and as many of you saw in the email that I sent around, there's what many consider to be a reproducibility crisis. And if we're trying to solve human health problems by doing science that can be reproduced and is a firm foundation in scientific concepts, then we need to make sure that we're all collectively working together to address this. So how do we reproduce and make sure that our science is reproducible? Well, we work on rigor and transparency. And today's speaker is somebody that I heard speak earlier this year at a pediatric research grand round. The way that, and he did not speak on this topic at all, but the way that he presented his science, I could tell, was very rigorous and very transparent, and I imagine very reproducible. So with that said, I'm pleased to introduce Dr. Marvin, who is a professor and a Georgia Research Alliance Eminent Scholar in Molecular and Cellular Biology. Also the Bennie H. and Nelson D. Abell Chair in Molecular and Cellular Biology at Georgia Tech. He is a director for the Center for Microbiodynamics and Infection at Georgia Tech as well. And he's near and dear to our hearts in pediatrics because he's one of the co-directors of the Cystic Fibrosis Center between Emory and Children. So his research interests can be summed up best, I think, at least I read this on the internet, by that he studies the social lives of bacteria. And I'm even going to, as a neuroscientist, I'm not even going to pretend that I know how to read the names of those pathogens. But as I mentioned, when I heard him speak earlier this year, even though this is not my area of expertise, he made it very accessible and not to mention interesting. So I'm really pleased to introduce Dr. Marvin Whiteley, and he will introduce the lab members as well.
All right, thanks. It's nice to be here. I have to admit, I've never given a talk like this, so we're going to have to see how this goes. Well, you know, I teach undergrads and I give a lot of seminars, but I haven't really thought a lot about how to express things, you know, sort of ideas about rigor and reproducibility. So hopefully, we're going to have a few things. I think we might have a few group activities here. You guys seem excited about that, but kind of like grads. Hold on one second. So I brought 3 people with me to dilute out the work and to sort of get a sense of, I thought maybe one thing we would do, we're going to go over some ideas. I'm going to give you a little outline before they introduce themselves first.
You do that, and I'm going to have to share your screen, because that's the only we didn't do.
Okay, sure.
All right.
So talk about 3 different things. We're going to talk about rigor and transparency, like what does it mean? And then we're going to talk a little bit about statistical rigor. I think a lot of you probably know about statistics, and you do statistics, and you do statistics till it says 0.05, and then you stop doing statistics sometimes. So I brought one person from my way. I have Dan Cornforth. Dan's a research scientist. He'll tell you a little bit about himself in a second. Dan's actually a math person, so it's a little scary. Gina in the middle is a postdoc in my lab. She's microbiology, and Juan is a grad student in my lab, and he's also microbiology. And before I let them tell you a little bit about where they're from and kind of what they're working on to give you a sense, the thing that we're going to spend a lot of time on actually is I thought maybe just, you can go read about rigor and reproducibility from NIH. It's pretty easy, right? Everyone's really thinking about this. If you don't use the word rigor in your grants now, all the reviewers write down that you didn't use rigor and you didn't use it in the right way. But I'm trying to help you, and so I think we can all figure out how to do that. But I'm trying to give you some practical tips if you're running a lab, like things I've learned over the years. I've had a lab for 19 years now. So I've had a lab for quite a while. And I remember when I started my lab, you just thought everyone was, you know, rigorous, right? I mean, you got some tech students, they gave you a bunch of money to start your lab, you're super excited, right? You're doing experiments, and you slowly start to learn, though, that especially young folks and grad students need lessons in this, because a lot of times, particularly in biology stuff, we're not so rigorous. So that's kind of what we're going to go through today. But Dan, you want to start out and just tell everyone who you are, and yeah, that one.
Hey, is this working?
Yeah, really close. Sorry.
I'm Dan Cornforth. I'm a research scientist in Marvin Lab.
Tell them about where you're from and where you went and all that stuff.
All right. I grew up in Texas. I did my grad work at UK. So I studied undergrad was math and PhD in evolutionary biology at the University of Oxford and the University of Edinburgh, Scotland. And I joined Marvin Lab after that, learned some microbiology. So that's still where I am. I'm a research scientist.
Hi, my name's Gina. I'm a postdoc in Marvin's
You're not a grad
lab.
student, right, Gina? Right.
I've been in Marvin's lab for about 2.5 years. I did my PhD at University of Wisconsin-Madison, microbiology, studying microbial ecology and evolution. And in my work now is still kind of in that vein, doing combination of bench work and kind of computational bioinformatics, sequencing sort of analysis.
My name is I'm a fifth-year grad student at Marvin's Lab. I did my undergrad research in biochemistry, in University of Texas at Austin. And my main focus is microbiology and developing tools to characterize spatial organization in microbes and cellular microbial interactions.
All right, so I'm going to start out, and then we're going to sort of tag team a little bit when I feel like they kind of have something to say that I don't. And that's really going to be a lot of just practical tips for running the lab, because I'm never in the lab, and I'm never there. I'm basically traveling probably a significant amount of the time. And so it's actually really interesting over the years how you had to set up sort of really sort of definitive and precise framework so that folks can actually work on rigor and reproducibility. So I'm going to tell you how we do that and hopefully get some feedback from you, because I don't want to just be up here the one talking. So we only have a couple of slides. I don't really need to do this, and Stacy did it, but I want to give you a couple of definitions. Did you get these definitions along? Use your microphone. Use your microphone.
The first one, rigor, since rigor is sort of like a general concept, I just got it from the dictionary.com, something like that.
Yeah, okay.
Yeah, like a combination
On
of-
your phone. Yeah.
on my computer.
Yeah, there you go.
And then transparency, there's different definitions of transparency, and this was scientific transparency, and I got it from probably like NIH or something like that.
But the idea of rigor is just then really what we're thinking about, hopefully reproducibility. That you can do something rigorously enough and then actually explain it to people in a transparent way so that they can reproduce. That's what we're trying to do. So how many of you have tried to reproduce something that doesn't work? Have you tried to reproduce something from the exact lab you're in that doesn't work, but that is now gone and you can talk about it? Right? I mean, so reproducibility is not just across labs, right? Reproducibility is in the same lab, right? You'll get someone new in, and they'll bring you some gel or something, this doesn't work. And you know, why doesn't it work? Didn't you get the protocol? Yes, I got the protocol. Well, let me see the protocol. Well, this is the protocol. It doesn't work, right? So it's even hard sometimes in the exact lab to actually reproduce. And it's really difficult across. What do you think the biggest sort of, they tell you about a cool psychology study where they actually have tried to reproduce a lot of the results that have recently. But why don't we reproduce a lot of each other's results between labs? We do that. Why isn't it common? How do you talk, Jeff? NIH doesn't pay for you to reproduce someone else's results usually, right? It's a waste of time. Actually, what Dan's going to tell you about is a group who have decided that in graduate education, early in your first couple of years, you actually have to reproduce someone else's study before you start your own work, right? With the idea being that that's sort of a way that you could actually go through and begin to reproduce that. But if we do all the experiments that we do with these really expensive animal models, we're never going to do this, right? We're not going to have the resources to do it. So how can you actually try to address that a little bit? And so I got Juan to actually go out and look into finding nice examples, not from any labs in Atlanta, just in case anybody was here, but from consequences when you do research with no rigor or transparency, and he was going to tell you a couple of stories.
Yeah, so this, I actually learned from the top example from my class. So basically in the 80s and early 80s, a really cool group between nature and science, and see who was going to publish the first structure of inherently the primal proteins, or that are like floppy, so the crystallography wasn't developed enough in order to characterize the structure of these proteins. And so science won. And basically the way that crystallography works is that once you have kind of like the main structure, then you can, like from protein, then you can, based on that structure, start build up more similar ones. But the hard one is to get the first one. So then they get the first one, and then they started publishing a bunch of them. Turns out that the first one was wrong. And Nature published like, oh, we got this not so similar protein, and it's fundamentally different from the other ones that have been published before. But if you flip it and along an axis, then you're gonna get the structure that these people found. And so it was a huge retraction.
So they took the first structure as the gold standard.
Right.
And then any data that came in that didn't fit that gold standard, they rejected it unless you manipulated it to get, and it fit that standard. And then you could make your paper if you changed it to meet. And then it turned out that that first structure was not right.
Right. And then they not only like showed that it was right, but they showed that how could you get it wrong? Like, oh, if you look at it like this, then it's understandable that all these people like. I think that was a cool example of like how thorough are you being on your experiments, and how thorough are you being in your analysis, like making sure that any possible aspect of your experiment and of your analysis is being taken care of. And then the second one, this is way more recently. This was happened in 2014. STAP cells is short for stress trigger acquired pluripotency. So this was a group that basically said that you could just stress cells, and after stressing the cells, they would induce, you would bring them back to pluripotency. Turns out that after a year, people could reproduce it, and then they were sending people from that lab to try to reproduce it, and basically nobody could do it. And then if you look at the Wikipedia picture of the person who published this research, she also got not only she was a postdoc at that lab, but she also, after an investigation, they found that even her PhD dissertation was, but she was copying and pasting from other sources. So this is another example of how transparency can basically ruin.
But I have a friend that actually worked on this at UT Austin, and she was really interested in this. But the big problem was, in the paper that was published, the methods weren't enough that, you know what I mean, they weren't precise enough, and they always thought they were screwing something up, right? Like, oh, I'm adding this wrong. They're not doing it exactly right. So they spent almost an entire year trying to get this to work in their lab, and they couldn't get this to work at all. And it was all because the methods were written sort of in a way that wasn't really, I think, well established that you could actually do the experiment. And Gina's going to do something in a group with all of you to see who reproduce something in a minute. All right, so you want to go, Gina? Get up if you want. You're not required to sit.
We already talked about rigor a little bit, but this is just and I pulled from a microbiology journal thinking about rigor on kind of the different aspects of it. So experimental redundancy, thinking about don't have one experiment that proves your main part of your paper, look in different ways, have the control, have the right number of replicates. We're going to talk more about error analysis, be intellectually honest about your work. Don't be afraid to acknowledge, if something doesn't match with your hypothesis, make sure you acknowledge that, make sure you acknowledge the work that comes from you that other people have done. And we'll talk more about probability and statistics too. And then another thing kind of is just within thinking about logical fallacy methodologies and making sure not that you're trying to prove your hypothesis right, but that you're also trying to prove it wrong, and you're not just assuming, like, if X, then Y, and if you find Y, then, oh, then X must be true as well. So kind of thinking through that stuff the right way. Really, a lot of this just comes down to being honest. When I was thinking about this, I remembered, I think this was, like, mostly popular in 2012, 2013, these overly honest math that people were putting up on Twitter. You can read them for yourself, too, but, like, reaction ran long due to extra time in World Cup games, or perfect Western blot exposure time, however long it takes to drink 2 beers and bitch about France with friends. This experiment was done with 7 chicks because that's how many I could fit in the tube before I got booking out, sort of thing. So, I mean, in reality, sometimes you don't have a plan and things just end up how you do your experiment. But we should have plans. You shouldn't just use the right the number of animals until you get significance. You should do a power analysis and think how many animals you need and have that plan in advance. You should have plans for how long you're going to do your incubation, and if you deviate from that, you should have a reason, and you should keep track of all of that sort of stuff. You should have an idea for how you're going to exclude outliers before you go in and collect your data, that sort of thing. So, kind of what Marvin was mentioning before is having good methods is really important for being able to be transparent and be able to reproduce things with math and across labs. So I pulled this method section from a Nature journal. It was the only methods for this. There weren't supplemental methods or something additional. So 2 viruses that they're putting into mice. And see what happens. And so I'll let you, can you guys all see that, or do you want me to read it?
So, why don't everyone read it, and then we can see whether you can actually reproduce this or
Yeah.
not. This just came out like a few weeks ago.
Read this through, and then friend or make a friend sitting next to you if you want, and you could talk through too. If you had to reproduce this procedure, could you do it? What else do you need to know? How could you make this better? What do you appreciate? What details do they have in there that you appreciate that they gave you?
So we'll give it a minute or so.
And one thing I'll just mention is, even though this is a mix of basic scientists as well as clinical researchers and outcomes, I still think that just kind of get through the nomenclature, and you could still apply this to other types of research.
Yeah, for sure.
Yeah.
We'll take steps through it in a second. I just want to see what everyone. So if you think who thinks this is good? We didn't pick good or bad, we just chose. So you guys think this is well written and just, did you go to the lab and reproduce this like exactly? Just like more than you guys. So, who thinks that it's well written and you can reproduce it? Who thinks couldn't? Probably kind of difficult, it seems like. Why is that? What's missing, do you think? First of all, who in here are trainees right now? Ever taught you the method section? Or do you just write it and hand it to your advisor? Is that what you do? Yeah. It's really formal training in this part, right? And then the advisor doesn't know what you do anyway. And so they're waiting on you a lot of times, right? I mean, they need you to do it. You're the one at the bench, right? So they're like waiting on you to write it. Like, what exactly did you do? So what do you think, what kind of precision do you think is missing in here?
Where they're from and how are they propagated?
Yeah, and that would be nice. Yeah.
video of the mixture of wild type and.
That's good, right? So, like, you do that? Yeah, you just randomly choose? Yeah.
Anything about the gradient, like how much of that,
Yeah.
percentage is.
That's right. How would you do the gradient, right? I think that's a big one, right? I mean, how would you go in and actually do that, right? What else is on, Gina?
Do you know how long does this experiment go for? I don't know what the inclusion criteria are for the animals or when you read from the study. Is that no.
Indicative of the kinds of, I mean, I'm getting older, right, so I complain about, like, I remember the old days, how good it was. You know, used to, before we had supplemental methods, you actually had to write the methods. Now you can throw them in supplemental, and a lot of people don't. I mean, some of you look if you want to repeat it, but no one really goes on, right? You get the paper, right, and you look at it, right? And I think this has been a big problem with methods, is we've dumped them into these supplemental sections now, rather than keeping them up front, right? Because this is the most important part. This dictates everything you're going to get. The journals have decided it's not important, and so we dump them in the bag. And we also always reference people and say, oh, we did it like we did it, and then they reference someone, and then they reference someone, and then they reference someone, and then you're back in the 80s, right, where someone did a centrifuge, gradient, and you're supposed to know that that's exactly what happened, but you know it's not, probably, right? And so I think that's a big thing. So I think a lot of the reasons why things are not replicated the way they, I think, it's because we don't know exactly what people did, and it really makes a big difference. Yeah.
Comment question. But the method to say so you can see the online supplemental version, right? Now, assuming that a lot of the genesis of that was, well, so it won't count against author's word count or print count or so forth, and that because to me this is like, right? They cut out connecting. Language is not good in methods, but for word count you cut out non-critical parts and leave out certain things and so forth. So, but maybe that, if you put it in the supplemental section, that you would eliminate the need to that, and so people could write, more thorough ones that didn't have a word count and so forth, and just put on it. But it seems, I'm assuming that maybe was some of the rationale for doing that.
Yeah, some of the rationale was to let people expand, right, I think a little bit, and then it just became available. It's why you need 10 supplemental figures to publish a paper now, right? You didn't used to need 10, and now they ask for every experiment, right, that even ones you don't need to do, they ask, oh, just throw it in supplements. I mean, and so I think that's very common. But, I mean, sort of Nature and Science started this kind of in the beginning, in the early, they would make I don't know if you guys remember, but the font was really small on the methods. And by doing that, it was like this idea that the methods don't matter as much, right? It's really the results that matter. You shrink the font, and then you finally just get rid of it, right? And rely on you to go actually look it up. And then I just don't think, all of you probably reviewed something in here. Do you review the supplemental as hard as you review the main grant? You're not critical generally. Some people are, but most people aren't, right? They'll let the supplemental go. Like, it's in supplemental, I don't care, right? It's main paper, right? And as an editor, I'm an editor of a few journals, and you see that a lot. Like, everyone just says yes to supplemental. There's this, when you review, probably all of you have said supplemental yes or no. I've never had a no. Like, everyone just says yes, because they look at it, but they're like, it's in supplemental. It's not that big a deal. But I think there's a huge problem, like replicating data, I think. It's just the idea of being able to explain it. Yep.
So what's the solution then? If the journals are not supporting good, rigorous, transparent method telling, are you saying that scientists should take it upon themselves as if they're reviewing?
Well, I think scientists should always take it upon ourselves, and I think that we should require it when we review, I think. And if you do that, then I think you're taking a step forward. They're going to do what we want them to do, right? And people are going to take a Nature paper with no methods, right, before they take some other journal, just because we have it in our head that paper is better, right? Even not a lot of times. Right? And so I think people really think about that, where papers go, and it's such a big part of our career that we think about those things. And those papers are really short, and people, I think that's why people do it. But I think we can solve a lot of the problems if we're just really rigorous when we review. I think that solves a lot of the problems.
In some labs too, they'll have on their lab website their protocols, like their standard lab protocols that they use for stuff. And I always really appreciate as a researcher when you have that too, and you can go back and.
Till they don't get tenure and their lab's website is shut down, right? Or they move labs, right, and they don't put it back up. I mean, this is why, like, we'll get to it in a minute, but putting data up, making data accessible, it can't be local, right? It can't be locally accessible. It needs to be accessible outside of the local. Yeah.
I think in taking your point about about Rick, I think that, I think it's shocking that journals aren't held to some type of, because in the end, the problem is it's a business. And so
yes.
It's a business, and so you have to cut out stuff that might be important, or clearly is important here. Things are now constantly being, invalidated from these major journals, but there's no consequence for them.
Yeah. That's true, but again, I think it is, but it's because of us. Nature exists and is going to help you get tenure because of us, nature and science, because we've overvalued those papers so much, and if you get that, we look at it and go, oh, this person must be good. Getting an R01, what does that mean, right? You get millions of dollars other places, you get an R01 and everyone all of a sudden thinks you're good. Right? Just all of a sudden, oh, that person's really good, they have an R01, they have 2 R01s, they're even, 3, they must be God. And it just, and it makes no sense, right? Those grants are hard to get, but lots of grants are hard to get. NSF's harder than R01. And if your work fits there better, right, and I think it's just a construct that's made by us, I think, that we do when you're on promotion and tenure and these kind of things. You do a construct. Hey, from anyone? Yeah, this was, Gina found this graph, right? Or was it or Dan? Yeah.
Yeah, so this was thinking about how do we make our data sets available currently. Sorry, I'm not picked on yet. How do we make our data sets available from our research? It was a study that costed of papers published in PLOS 1 from 2014 2016, and just looked at their data accessibility statement and saw where did it say their data was available. So you can see people put a paper, they put it in a repository. Some people said that access was restricted. I think that was sometimes clinical data where they said that, they couldn't de-identify or there was no way to do that. Some people have combinations. Some people, like a few percent of people had just data available upon request, which is the best thing to do. Said when they did this study too that there were 2 papers, I think, of these 3 years, where it said, like, in the published version of the paper, like, data is available, and then it was just like XXXX. And so even that sponsor review got through the final editorial checks and everything and was up online. So don't be that person, I guess, and we all try not to be that person. But I agree with what Marvin said, that, having the data somewhere where other people can access really the raw data, they can reanalyze it in whatever way they want, makes a huge, and honestly makes your work more likely to be cited and included in other studies and to, like, add more to, our science base than if you don't do that.
This was something I got Gina to pull, and this is, I'm not quite old enough to complain about this much, but my more established colleagues do complain about this, and you always say that was done in the 80s. And it'll be some science paper, right? And then you look, you really realize that a lot of people don't cite previous work a lot, right? And they feel like it really downgrades sort of how cool their data is, right? Whether it's going to go to a really good journal. And we actually had a talk at Georgia Tech a few weeks ago that is very indicative of this. Like, something was discovered, but it was really discovered in the 80s. And they had worked a lot of it out. They didn't have the genomics to actually work it out sort of to the resolution. That was the same now, and it was still really cool work, but never was this actually mentioned throughout an entire seminar. I think it happens a lot. One reason is because most things are online, and if it happened before the 80s, no one in my lab knows where the library is, right? So you can't go get it. And so you actually don't go look anything up. But there are actually some papers. I wrote a review many years ago that I went back and read some papers from Pasteur and Koch in the 1870s and 1880s. And a lot of the things that we were talking about and had published, and there was a recent PNAS paper, they actually shown Now, you know, they didn't have the molecular data right, but the fact that you could actually do these really interesting experiments. I mean, Pasteur screened for antimicrobials in the 1870s, things that kill anthrax, other bugs that do, and found bugs. In fact, he found the bacterium that we work on in my lab that kills bacillus, and that it was in the supernatant and all of these things. But of course, we don't read those papers anymore. But there's a lot of ideas, I think, that have been sort of around a lot, but we don't really know about it. So if you're young, then you should always go look. But if you're in grad school, I think it's always good to go to do a deep dive and see what people know before you start. This is you, Dan. We're going to make sure this doesn't go too long because this is very mathy. Dan likes math, so he wants to talk about statistical rigor. But some of this is kind of funny, so.
Oh, math. So, yeah, so, y'all hear me? This is following in line with what was just said about how useful it is to actually publish raw data in addition to whatever you yourself might find useful or whatever you yourself might find validating to the case that you're trying to make. So as an example of data points that I did, typically people only, at least in our field in microbiology, people only typically present the data as a bar chart. These are the same data that can have very similar summary statistics in terms of what it would look like, but they look completely different from one another. So they're saying different things in these different cases. So for instance, whether it's an outlier that might drag the data one way or the other, it's bimodal. So in D, you can see that data are actually clustering by axis of it. It might not be interesting to you, but it might be interesting to someone else. And by reporting summary statistics, you not only make it difficult to reproduce what you're saying, but it's valuable for people in the future. So typically, it's better to just be upfront with all of your data, and if it's possible, publish your raw data. So I do a lot of kind of big data analysis on transcriptomes and genomes and this kind of thing. So we publish everything that every time that we collect the data, every time that we sequence something, we always publish that with it. And journals aren't increasingly requiring that, but sometimes if you're reviewing, you have to fight with people to actually publish the raw data. In particular case, I was involved in reviewing a paper, and this author was very, very hesitant to actually give it in. I don't think that their conclusions were actually justified. Just making the point a little bit even stronger. So this is the famous mathematician who came up with this quartet, which is basically 4 different data sets have virtually identical sum distribution. So the mean of the x-axis is all the same, the mean of the y is the same, the variance of the x is the same, the variance of the y is the same. The correlation coefficient between them is the same. But you can look at them, you can tell there are entirely different things going on, and probably entirely different processes that underlie what you're seeing. So it's important to actually show the data.
You have any else? Yeah, one more, yeah.
So I'm not going to get too much into detail.
This is Dan's big pet peeve, so we're going to have to cut it off.
Well, yeah, so p-values. So the idea of p-values, you basically are trying to say how likely something would have occurred by chance. In the assumption there's no relation between the data or no relationship to the variables that you're measuring. And so it usually gets reported at cutoff score. So in our field, people usually are about 0.05. Any thing that has a p-value of less than 0.05, people get happy. You can publish it then. But if a little bit over 0.05, it's worth it. Obviously, this is a completely arbitrary cutoff. Different fields have different cutoffs, but generally 0.05 it will stick to for no reason. But this is the number of big problems. So the two I'm going to talk about is publication bias and what's become the p-hacking. So publication bias is just the idea that, like I said, people won't publish if they're getting below 0.05, and they will if they get above it. And so people have actually done these meta-analyses of published literature. They see the signature that you see in fields that have this problem. And it's just an example, but it's been studied in the several fields. And so the black is what you expect.
What does frequency mean?
So the frequency of that reported p-value. So these are called p-curves. So you take a bunch of different studies and you look at the reported p-values for all those different studies. And what you see that you see an enrichment basically right below 0.05. And that's black. Or sorry, there are cases where you see that that enrichment. The black cases is what the statistics tell you should actually be happening. So if you look at a bunch of different studies, what you see is this sharp cutoff at 0.05, and no one's reporting anything else. And this is kind of indicative of a publication bias, which is the field in a number of ways. But what is that in a second? Example. Second is
Because this first point here is, red point here, this is 0.06. Basically, if you're 0.055 or 6, don't publish it, right? Never see papers that have those p-values, right? But 0.05, you publish it, right? It's a huge, because you wonder what's out there, right, that hasn't been published that was 0.07, and that just means 7 out of 100 versus 5 out of 100, right? It's not really that different, right? And then I think what it causes is a lot of people do the same experiments, right? Right, because these aren't published.
yeah, if you're sitting at about 0.05, if you do the experiment again, there's a 50-50 chance you'll be above or below that. So really, it's a silly precedent, but it's stuck to so much in public. So here's another big problem that's called p-hacking. So this is known as basically different types of p-hacking. One, we were just talking about in the office earlier today, that this study actually said in their methods that they continued to collect data until they got a p-value less than 0.05. So in other words, not enough yet, not enough yet, not enough yet. Oh, it's good to publish. They didn't, this isn't fabrication that I'm talking about. They think that they're doing it right, but they're not doing it right. And like I'll say in the next slide, this causes big problems. People do these p-curves. This is a picture of p-hack. So right above 0.05, you get a big enrichment in publications that have that reported p-value. Another really common one is that people will they'll have a lot of variables they're looking at, and they won't control for how many tests that they're looking at. Because this is just saying, by chance, if you do one study, if you do one comparison, that's just by chance you're going to get this value if there is no relation. It's not saying that you can't find correlations if you look at 500 different variables correlated with each other. By chance, you're going to get a lot of that. And if you don't go for that, you end up with a lot of false positives.
It's odd though, right? I mean, so you look at this black line here, I mean, this is the number of studies that use these p-values, but all of a sudden at 0.05, you get a lot more studies there than you would expect, right? And it's because I've had this before with someone, and we asked them, well, what test did you use? And they said, whatever gave us 0.05, right? I mean, you don't see that with everyone, but you do see that sometimes, I think, and I think it's inherent in biology, at least our field a lot, because people aren't really, don't have statistical backgrounds. We're lucky here at Emory that there are people you can go to, right, that you can actually ask about. Another funny one.
So, just kind of a case study of what can go wrong and what has gone wrong when people do this. It's been looked at in psychology because there's a psychologist who decided this was an issue. My guess is if you looked at almost any field, any biological field in which error bars exist, if you look in physics, you don't have to worry about a lot of these issues. Or right. But in biology, there's noise. In almost any of these fields, you're going to end up with this problem. I'm not especially picking on psychology, but basically they went back and with a lot of collaboration, 270 authors, they decided this author decided to reproduce a lot of these studies in experimental psychology. And what they determined was that about a third of them actually worked the second time. So like I was saying, it's hard to actually, there's little incentive to actually do this because you get money for showing something the second time around. But what they is, and there's a couple schools that are continuing to do this, they've made it kind of part of their graduate curriculum, that as part of your training, you have to validate a previous study. But the left, this is the distribution of p-values in the, I should say, this work was all done in collaboration with the first authors. So they reproduced every single step of the published method. So I don't think that any of this was fabrication or by something, but I don't think the majority, I don't think what we are seeing here is a signal of fabrication. I think this is p-hacking. So what you see is the distribution of p-value. When you see the cutoff, the dashed line, below that to be 0.05. And so the original study that Marvin's pointing out now is there, and then when they reproduced it, they got this other one. So very few of them are actually cutoff. Again, trade cutoff, but if you're getting p-values way up there, it's not good. And on the second one, these are the actual side effects. So you can have a p-value that's very good, but the size of the effect might not be huge. Say, gene expression might increase by 50%, but you might be really, really sure, so you get a good p-value on it. So also, the size of the effect, from the first time that they actually did the experiment. So I know there's other fields that you see the exact same thing, so this is the problem with kind of rigged system until you get exactly what you want. Questions? Yes, please.
So is the solution then to consult with a biostatistician during study design and do a power analysis or calculation?
Yeah, I think that's a big one. I think it's also best if you can to stay going to test before you look at the data, instead of, doing what happens, which is that people just are trying to get a particular result to publish. Yeah, it's best to decide a priori. If people do this, if you want a one-tailed t-test or two-tailed t-test, you should know what you're going for before you do it. Don't just say the one that's less than 0.05.
Finding, so you have the people reading the results, they're deciding what to include or not, are not the people who set up the experiment, or the things.
Maybe a dumb question. So how often, as an editor, have you requested from others to change the statistical methods of the test they have used because it's inconclusive or it's misleading?
Yeah, the question is, as an editor, how often have I asked someone to actually change the statistical methods? All the time. I mean, I think all the time. Well, a lot of journals now actually have comments from reviewers. You guys have probably seen statistics. They'll ask you about the statistics. Were they good? Were they bad? Did they do the right stats? And as editor, you can rely on people to actually look at that and do that. But a lot of times, it's just correct, and I think that's definitely incorrect. They still may be valid results, but the tests are incorrect. So I do it a fair amount. I think a lot of people do that. Anything else? I just wanted to tell you, what do we do, right? So I've been in the game for a while, and I feel like we've always been very rigorous. And I've been doing a lot of these practices a long time, but I wanted to tell you a quick story about someone I know very well who made me even more worried about rigor in the lab. And that was a faculty member who was an associate professor, and he had a fairly large lab, about 8 or 9 people. And he had actually rotated in my lab, and rotated in his lab, and chose his lab. I was very upset, but then I'm not now. And he meets every Friday with people from that. You bring your notebooks in, and he goes through it with you every Friday. I don't do that, right? But he does that. And what happened is he had someone in the lab who just fabricated an entire PNAS paper, and the notebooks up, they look like that, ran experiments in the lab, and was running gels and running all kinds of things, but just made up. And it hurt his career. I don't know how he would have caught it. I mean, she literally was in there doing experiments and doing all these things, and so it was really difficult. To come back from that, right? Once you get that, it's a really hard thing to come back from. And so, I think that's a rare example, but there are times that this happens. And so we do a few things in our lab, especially since I'm not there a lot, to try to people think about rigor anyway, right? I don't know that you're always that's a very extreme example I told you, and I don't know that you're always going to be able to that or be able to catch that. But here's a couple of things that we do in the lab our daily lab work, something that in my lab we have peer reviews and writing groups. And I'm going to let them tell you a little bit about those in a minute. But these sort of came about as a way to try to have internal rigor between the students and the postdocs. And we actually do it between labs now, so there's a couple of labs at Georgia Tech that now come to these, a few people from their lab. And how they work, and then you can tell, then if you have any questions. So what we do is I split the lab into groups of 2 or 3. Each month, one person tells the other ones what they're doing with their lab notebooks, and they tell them exactly what they've been doing, the experiment they've been doing, any problems they might have. This does a couple of things, because it sort of lets people know what you're doing, right? As your lab expands and you get larger, a lot of people don't know what each other are doing, so it allows that to actually happen. It also allows people to be very critical of each other and be critical about their own work. And then the rest of the group actually asks questions, and then they write a report that comes to me about that person, and like what they're doing, and what experiments they've been doing, what they should be thinking about, right? Was the data analyzed correctly? Do you think they should analyze it a little bit of a different way? But then it turns around, because the next month they get to review you. And not to be punitive, right? I think it's more of the idea that you get an example every month of being rigorous, like that we are going to hold you to account for what you do. And they have to say they like it, but yeah, isn't it great? In your hands. No one has any comments about it. You didn't do it in your previous lab, Gina. Say how cool it is.
It's great. No, it's good just to talk to other people in the lab, and Marvin often will set us up with somebody who, isn't the person in our office, isn't the person like working on a similar project to us, so you get a little bit of an outside feedback on stuff, and you know what other people are doing. And I'm working with a younger grad student right now in theory vows, and so we were just talking about, like, well, what format do you use to keep your lab book and that sort of thing, which I think is really useful.
Especially the way that we've been interacting with people from other labs. I think that just being office, just being in another lab, just gives you a different mentality on how you like attack different questions, and how you do certain things, and it gives you like a review of your own work. And
you guys have thought of all you can think of it.
What?
You've thought all you can think of, I know, right? Right?
Yeah.
Well, we only have a couple of minutes though, so I'm going to go to the writing group. So the other thing we actually do in the lab is we break everyone up into writing groups. And it's usually broken up in, and I don't know, maybe these things, I stole these from other faculty when I was younger, so you guys can steal it from me if you think it'll work for you. But what we do is we have people at different levels, about 3 or 4 in the same writing group. Before anything gets to me, it has to go to everyone in your writing group, and they have to okay it essentially before it comes to me. But it also allows this young grad student that I was working with, for Gina to see the materials and methods, right? And go, this isn't exactly how you write it. You need to, so training can actually come from, I think, the lab rather than only from one person at the top. Because ultimately, as you move along in your career, generally you have less time, honestly, and you will just less and less and less time. And so it has to be more of a kind of internal thing. I think we have a couple of minutes left. This is about all we had. I don't know if it was informative. Some of your faces seem to think it was, others not. Is there any questions? Yeah.
So I love this writing group concept. I'm just wondering what impact it has on authorship.
So authorship for me is very easy. You have to have an intellectual contribution, or you can physically do experiments. We don't put everyone on papers, because it dilutes that first person who does it. So you do me on papers for reading. You're being a good lab citizen, and you're actually. So yeah, I might have anyway. You don't get beyond.
But it's probably important to establish that up front, I imagine.
Well, yeah, because you don't have to come to my lab. Yeah, because that's what we do. I mean, I think you see these papers with 20 people on them sometimes, and you wonder what 13 or 14 of them did. And so it's nothing.
I'd say that it gives people practice, like in the groups, to kind of see a paper come along. They only have their own experience. They have everyone in the writing groups. They can see that at first was hard to read. They can see why it was hard to read. They can kind of see it getting better as
you know, you also will benefit from this too, right? This goes with grants in my lab. Everyone who has a fellowship in my lab goes, you know, you go through this process and it helps, right? I mean, I think authorship for me is significant intellectual contribution or physical experiment. Is there anything else?
How long can a report say?
Depends. Some people half-assed it, and so it's like, you know, too much. And then some people write a whole page. I don't need a whole page, right? The goal for me is just how's that person doing? Because especially if it's a grad student and I've gone a lot, you know, it's hard to know, is this person moving along fine? Do I need to have special attention on them? Is Dan or Gene, somebody really advanced in my lab, worried about them, right? You know, from the last 3 months. It gives me a really good report for them. And so it just really depends, I think, on how that interaction is. Yep.
You mentioned before that the seminar where you got the sense that you're very rigorous. Could be an example of that as well? I think that would be really useful to see how you present your results or in a seminar present your findings that matches with some of these things we're talking about.
So I think the biggest thing is when you do your introduction to whatever you're talking about, don't leave other people's work out. Like, say what everyone's done. A lot of people only say what's come from their lab. Say what everyone else has done, right, and where the field actually is. And then when you get results, I think you say things about, oh, well, you know, we did this because of this. You know, like be really, I think people aren't explicit because I think anytime I don't know, as humans, we know when we're trying to hide something. You know, if you ever feel like that, you should fix it. Right, just be completely open about it. People, I think, trust it probably more.
Yeah, I'll tell you from my perspective, he did not do any of the, well, you really can't see this figure, but trust me, this is what it says. And really just explaining what the axes mean, what he measured, and so not just giving conclusions, but walking through the data. That's what really impressed me during your presentation.
I would say, in addition to this, anytime anybody gives a talk, has to see it, and data has been fundamentally essential for surviving grad. Because we go thoroughly over, okay, you need to, yeah, exactly as you said, you need to, oh, what are you saying? Point out what you're saying. Oh, okay, this doesn't make sense. We go thoroughly over each and every talk that anyone gets.
Well, I mean, it's awesome.
I think I fairly good in giving a talk, and it's because of that.
Yeah.
All right. Okay. Thank you, and thank you. And next month, we'll be doing a panel discussion with case studies on mentor-mentee relationships.
You said that. That's okay.
No, it's okay.
See your slides on peer reviewing. I wanted to see the peer reviewing part. Yeah. And I was wondering, I forgot to ask, it has something to do with Rigor and reproducibility and things.
I think it has a lot to do with rigor.
No, not this. My question.
Oh, okay.
When you have many people in your lab, do you have some sort of a code of conduct